Hello and welcome to the TYBSc students. After completion of the first unit, that is Mendelian Genetics, we are going to start a new topic, that is linkage and recombination. In this particular topic, we are going to study the linkage and recombination or crossing over with their definition and significance, and particular the concept of the linkage and recombination. And the last bit in this particular topic is mutation. So similarly, mutation we are going to study with their definition, different types of mutation, and the significance of the mutation. So let us discuss about this second topic or second unit, linkage and recombination. So let us discuss in detail which subunits are included in this unit second, linkage and recombination. So the first subunit is linkage, which we are going to discuss that its definition, linkage groups, types, coupling and repulsion phase, and the significance of this particular linkage. The second subunit is recombination or crossing over. So the recombination, we will see its definition, its types, mechanism of crossing over, and lastly its significance. The third and sub last is mutation. So about the mutation, the definition, spontaneous and induced mutation, these main types we will discuss in detail. Then types of the mutagens, the two subtypes are there, physical mutagens and chemical mutagens, and lastly the overall significance of the mutation. So these three subunits are there. So let us see the definition of the first point or first subunit, that is linkage. So it is defined as when two or more characters of parents are transmitted to the offspring of few generations, such as F1, F2, F3, etc., without any recombination. They are called as the linked characters, and this particular phenomenon is called linkage. So there are many definitions which are provided in the particular notes which I have given to you, and the linkage is the consequence of the concerned genes being located in the same chromosome. This particular deviation from the Mendelian principle of independent assortment. So you all of you have know the law of independent assortment. So this particular linkage is deviation from the principle of independent assortment. Here we can see a particular linkage map of the Drosophila melanogaster described by Thomas Hunt Morgan. This was the first, so we can see this particular genetic linkage map. This was the first successful gene mapping work and provides important evidence for the chromosome theory of inheritance. There are different views of many scientists about this particular concept of linkage. let us see the Thomas Morgan's view of the linkage. So Morgan in 1910, while working on the particular insect Drosophila melanogaster, he stated that the particular linkage having two aspects, that is coupling and repulsion. He defined the linkage as the tendency of genes present in the same chromosome to remain in their original combination and to enter together in the same gamete. Mendel's laws of particularly the Mendel's law of independent assortment is applicable to the genes that are situated in the separate chromosomes. When genes for different characters are located in the same chromosome, they are tied to one another and are said to be linked. Here, this particular sentence or these particular sentences explains that how this particular concept of linkage is different or not following the law of Mendel's law of independent assortment, and they are inherited together by the offspring and will not be assorted independently. Thus, this particular tendency of two or more genes of the same chromosome to remain together in the process of inheritance is called linkage. Now we turn towards the second view of the scientist William Bateson and Reginald Punnet in 1906. They, while working with the plant sweet pea, that is Lathyrus odoratus, they observed that the flower color and pollen shape tend to remain together and do not assort independently as per the Mendel's law of independent assortment. When two different varieties of the sweet pea, one having a red flower with round pollen grain and other having blue flower and long pollen grain, were crossed, the F1 plants were blue flowered with long pollen grain. Here the F1 individual is blue flowered with long pollen grain. Blue long character were respectively dominant over the red and round character. When this particular blue long which is heterozygous hybrids were crossed with the double recessive, that is red and round. This particular red and round, this particular blue long is crossed with red and round, the dihybrid test cross. The red and round is the homozygous. So the expected ratio of this dihybrid test cross is 1:, this particular expected ratio is 1:1:1:1. But Here the observed ratio will be different. Here we got the observed ratio with the particular percentage frequency. The observed ratio is 7:1:1:1:7 here, in which the ratio shows 7 individuals of blue long, 1 individual of blue round, 1 individual of red long, 7 individuals of red round. So from this particular above results of the test cross, it clearly indicates that the parental combinations blue long and red round are 7 times more numerous than the non-parental combinations. Bateson and Punnet suggested that the genes such as capital B and L coming from the same parent tend to enter the same gamete and to be inherited together. This is also particularly called coupling. Similarly, the genes B and L coming from the two different parents such as capital P, capital B, capital L and crossed with small b, small l tend to enter different gametes and to be inherited separately and independently. This particular concept is called repulsion.